Tonight we have with us Dr. Melissa Neuman, and she's going to talk about the, well, you can see it up on the board. I'm going to abbreviate it, some of the status of white and Pinto abalone in Southern California, and some of the efforts to restore these populations. Dr. Neuman is the Fisheries Abalone Recovery Coordinator for the National Oceanic and Atmospheric Administration's National Marine Fisheries Service. She has served as a fishery biologist with NOAA since 2002. Before that, she was a postdoctoral fellow with NOAA, and also before that, she was a postdoctoral fellow at Rutgers University. And when she was at Rutgers, she served as the assistant director for the Rutgers University Marine Field Station. In 2011, Dr. Neuman received the NOAA Fisheries General Counsel's Award. She's the author of a number of papers on the endangered status of abalone. She got her bachelor's degree from the University of Pennsylvania, her master's degree from Rhode Island, and her PhD from Rutgers University. So she grew up in New Jersey, and as we were saying over dinner, lots of people want to get as far away from New Jersey as they can. No, that's not true. That's not true. There are many beautiful parts of New Jersey. And you just don't see them when you drive the New Jersey Turnpike. But she went to the Isle of Shoals Laboratory in New Hampshire, and that was for her one of the transformative experiences. These National Marine Labs and biological field stations play really important roles in this nation. They're part of the research infrastructure, and they're the places where many people go as undergraduates and they get hooked on field biology, whether it's in the ocean or in terrestrial environments, and they all deserve our support. And this aquarium has been very involved in trying to elevate the perception of these institutions and why they're so important. Please join me in welcoming Dr. Melissa Neuman.
Thanks so much, Jerry, and thank you all for having me here tonight to talk about my favorite endangered marine critter, the marine snail known as abalone. Today I'm going to be focusing on the challenges and progress associated with trying to determine the status of two of the rarest species of abalone that occur in Southern California, the white abalone and the Pinto abalone. I'm going to give a little bit of a synopsis here on what I'm going to talk about today. First, I'm going to give some background about the cultural importance of abalone, the fishery collapse of abalone in California, some of the agency protections that came into place following the collapse of the fishery. Then I'll move into talking about survey approaches, using remotely operated vehicles, scuba surveys, in situ time-lapse cameras, and acoustic tags. The reason why survey approaches are so important to the world of abalone, and in particular white abalone, is because we still know so little about what factors in the wild are important for this species. And a major prong of our recovery efforts for white abalone involve captively raising white abalone, and then taking those captive raised abalone and placing them back out into the wild. And in fact, here at the Aquarium of the Pacific, you can see white abalone. They are one of our partners in helping to captively propagate white abalone along with 4 or 5 other partners here in California. So if we are to know where we should place these animals so that they have the best chance possible at surviving, we need to survey their environment, find out where they are now, what kinds of habitat features seem to be good for them, so that we can select those outplanting sites in the future. And of course, we want to know about the status of the species in the wild, because there are some remnant animals out there, and we need to understand how they operate and what allows them to continue to survive. So these approaches are all very important, and they each have their pros and cons. So I'm going to spend quite a bit of time going over the approaches that we have taken over the last decade. I'll talk about the results and what we've learned in terms of the demographics of wild populations, their behavior, and then, of course, next steps. I'll summarize the information, and then I will acknowledge all of the wonderful partners and collaborators we have who are working towards recovering abalone. So when I said marine snail, who thought cute and cuddly? Anyone? Well, for those of us who work with abalone, we do think of them as being cute and cuddly. And this fantastic picture by Kristen Aquilino, who works at the Bodega Marine Lab, shows you these sparkling little green eyes sitting at the end of eye stalks, and these cute little faces. So for those of us who work with them, yes, we do think they're cute, but that's not what drove them to extinction. What drove them to near extinction is, oops, okay, what did I do? Uh-oh. There we go. Okay, got it. This muscle right here, known as the foot muscle of the abalone, is what many people who fished for them historically were after. And that was part of the reason that their numbers became so low in the wild, this desire to eat this foot muscle. So a little bit more background information about abalone in California. We have 7 species along the west coast of North America, ranging from Alaska all the way down to the southern tip of Baja California. And all 7 of these species occur in our backyard in Southern California. We are in a hot zone. From Point Conception down to central Baja California, we have all of these species. They're in our backyard. We can't let them go extinct. I only have 5 of the species pictured here on the slide because these are the 5 species that the National Marine Fisheries Service takes the most interest in. Because of their endangered status, as is the case for black abalone and white abalone, or because of their status on the species of concern list, which is a list of species that NMFS is very concerned about, but we know so little, we're not exactly sure what's happening with the trend of the population in the wild. And they are the Pinto abalone, the green abalone, and the pink abalone. So I'm sorry if this is a bit of a review for some of you, but again, just to provide you with a little bit of background information for those of you who do not know, here's a little schematic of the life cycle of an abalone. Abalone do have separate sexes. Males and females release sperm and egg into the water column, and in a matter of hours, fertilization occurs. Roughly 24 hours later, as these different stages here are floating in the water column, a trochophore larva emerges from the egg. The trochophore larva is feeding off of a yolk, so it is not a free-feeding larva yet. And approximately 24 hours after that, the veliger larva forms. This is when the larva starts looking more like an abalone. Its shell is forming and its foot is forming. And then about 2 to 3 weeks later, depending on water temperature and a variety of water quality parameters, settlement occurs. And you have this early juvenile abalone settling down to the bottom of the ocean floor. Within a matter of months, this early juvenile becomes a full-on juvenile, and it begins its benthic post-settlement phase of life. At about 4 to 5 years of age, we think in the wild, males and females will become reproductively mature. It can occur much earlier in laboratory settings. We've learned that from our captive propagation program in the case of whites. But in nature, we think that they mature at about 4 to 5 years of age. And I will just stress the importance that because of this benthic lifestyle and because the length of life of the sperm and egg in the water column is so short, it's important for males and females to be close to one another in order for reproduction to be successful. So I mentioned that abalone were culturally important in California. And this goes back to thousands of years ago when Native American people living in the area relied on abalone for protein. And it was a constant and steady source of protein for them because they could dry the meat and they could keep it for very long periods of time. unlike other sources of protein that would spoil. And the evidence we have for this, of course, is the abalone shells that we find in these middens or garbage piles that are spread throughout our coast, but they also extend to inland areas where archaeologists have recovered shells as far east as, I believe it's mid-Texas there. So what were the Native American people doing with these shells? Well, we think they were trading them. Not only were they important food sources, but they were also incredibly beautiful. And the shells had a lot of utility, either for being bowls to hold food, to hold water, and also the mother of pearl was very, very beautiful, and they would make bracelets and all kinds of jewelry that they would trade to inland tribes. So abalone played a huge role in the cultural lives of Native American people. And the next few slides are just a couple of examples of an archaeological find, a recent archaeological find, where an abalone shell that was plugged with tar, the respiratory pores here are plugged with tar so that it could hold liquids without leakage. And these beautiful little ornaments made out of mother of pearl, we're not sure what for, but it's pretty incredible to think that this kind of time and energy was spent on manipulating the mother of pearl. Here's an example of a fish hook that came from the mother of pearl of an abalone shell. So there were all kinds of uses among the Native American tribes. So fast forward a few thousand years, and European and Asian settlers to the West Coast had an insatiable desire for abalone because of the way it tasted. So at this point in time, it was still valued for the beauty of its shell, but mostly it was valued because of the food it provided and the taste. And also with European and Asian settlers came this increased technology and tools to dive deeper, dive longer, and refrigeration on boats, and all of these things allowed people to really wipe out populations without thinking much about what their life history looked like. I forgot to mention in the life cycle slide that I showed that abalone we think can live up to between 30 and 40 years, potentially producing offspring, successfully producing offspring only once a decade. So with that kind of life history, you can't have this kind of fishing effort and expect things to stay the way that they are in terms of productivity. So as a result, we fished white abalone to near extinction. This is a plot here in the lower left-hand panel that shows you the year from 1960 to 1992. And here is thousands of pounds of abalone that were collected. Oh, I'm sorry, I think I have the wrong. Well, this is the recreational fishery. But either way, you can follow this trend, whether it's recreational fishery or whether it is the commercial fishery up here in the top right panel. And you can see that there was a period of about a decade through the 1970s into the 1980s for the recreational fishery when white abalone were intensely fished. And at the same time, this line here on this bottom plot shows you the value of white abalone over time. And you can see that the value of abalone stayed high. So this contributed to their continued decline. And the same pattern was seen for Pinto abalone, the other species I'm going to be talking about today, where an intense period of fishing over a decade nearly wiped out the population. As a result, the state of California and then the National Marine Fisheries Service listed abalone species in the state as endangered, either federally endangered or the equivalent of a state endangered category, because all fishing was closed. And we developed plans at National Marine Fisheries Service, at the state of California, and also abalone up the coast in Washington were not doing well, and in Canada, Pinto abalone were also not doing very well. So all of these state and federal governments developed plans. And in all of these plans, there was an emphasis placed on assessing the needs of these abalone in the wild through assessments and to continue monitoring their status. So with this in mind, we charged forward with our goal to better study these two species in the wild. We work very closely with our team of remotely operated vehicle specialists down at the Southwest Fisheries Science Center. Kevin Stierhoff leads this group along with others in La Jolla. And starting in 2001 or 2, when white abalone was listed as endangered, this remotely operated vehicle was used to search for white abalone in very deep water. The advantage of using a remotely operated vehicle, of course, is that there is no bottom time and you cannot run out of air. So the ROV, or the roving surveyor, was able to move into deeper waters where we were aware of some remnant populations of white abalone and look for those animals and try to follow them over time. There's a high-definition video camera on the ROV, a light array that allows you to see, and a bunch of cool little things that allow you to maneuver the ROV pretty well. Now, one thing that it doesn't do very well, it does take fantastic pictures, but one thing that it doesn't do very well is maneuver through kelp. So one of the limitations of the ROV is that you cannot be in shallower areas where kelp is extremely thick and abundant. Otherwise, it gets entangled. So this particular tool is fantastic for searching large distances or long distances. You can do a transect that's up to 600 meters, and you can actually drive it in a straight line. So you basically know where you are on the bottom, how much distance you've covered, and your field of view is about 2 meters. So the nice thing is you can get a measure or an estimate of density, and then if you multiply that by the amount of habitat you've searched, you can get an estimate of the total population. So this has been a great tool for us, and we've used it at some of the offshore banks and more remote places where we really can't do scuba surveys. Now, as I mentioned, there are some limitations to the ROV. So step in your scuba divers. And what they're really good at is covering smaller distances in water in habitat that's a bit more complex. And typically what divers will do is drop an anchor, set out transect tapes to either side of the anchor, and survey these transects looking for abalone. And when an abalone is found, the nice thing about laying out this grid is you can figure out exactly where you are on the ocean floor. And then you can start searching for nearest neighbors, and you know where they are. So there are some definite advantages to using scuba as a method for surveying. Now, here's the final method, or the second to last method that we've used to look at abalone on the ocean floor. And this is an in-situ time-lapse camera. And I'm going to start a little movie here. This time-lapse camera was developed by a citizen science partner who is an engineer down in La Jolla. And he and his partner built this camera and the frame. And believe it or not, these cameras are incredibly good at staying on the bottom. I don't know how it happens. I don't know why they don't move, but they're weighted, of course. And we direct this camera on an abalone, and it takes a photograph every 10 minutes. And then you can pick up your camera at the end of 2 weeks or as long as a month after you deploy it, and you can download your photographs and speed up time. And you can get a sense by matching that information up with environmental data. What is an abalone doing over the course of day-night, during different moon phases, when there are huge storms? What are they eating? When are they eating? Are they spawning? So this is an incredible tool for getting a look at the very fine scale behaviors and needs of abalone in the wild. Okay, and the final method that we have just embarked upon is putting acoustic tags on adult abalone and setting out an acoustic array of receivers to detect the movements of Pinto abalone over time. One of the big questions that we have about abalone is how much do they move over time? Do they move more during certain times of the year? Under certain conditions might they move more? And we know that we have a very rare and patchy population of abalone out there. So if you do have an abalone that is separated from a nearest neighbor, do they have the ability to find a neighbor during the time of year that they should be spawning? Well, Pinto abalone was a great model for testing this, and using this acoustic array was a great tool because we had a group of Pinto abalone, that some of which occurred in small groups and some of which were solitary. So we set out this array. You can see here that the array spanned an area of about 100 meters by 70 meters, but the detection area for the array is the area that you see enclosed by all of these circles. So we've been tracking Pinto abalone since the fall, and I'm going to present some results from that experiment today. And here's just another method slide showing you what one of these acoustic receivers looks like on the ocean floor. They're just moored to the bottom with concrete, and this is a piece of rebar here, and this is the actual receiver. So this is the instrument that's receiving the signal from the tag on the abalone. So now I'll move into the results section of the lecture, and I'll start first with some of our ROV results. So each of these yellow dots in the bottom left panel here shows you where a white abalone was detected on this offshore bank. These black squiggly lines are the track of the ROV. And the different shades of blue show you 3 different depth zones that were surveyed. As I said, you have a lot of maneuverability with the ROV. So what our ROV scientists did was they made sure they drove the ROV within a depth strata so that they knew when they were sampling in the 30 to 40 meter depth zone that their ROV was running a straight line only in that depth zone, and allowed them to do an analysis that was depth stratified. So taking a look at the lengths of the animals, the other advantage, by the way, of using the ROV is you are collecting video during the entire time that the ROV is roving. So even if you don't see an abalone on the first pass while you're actually operating the ROV, you can take those videos back to the laboratory and review them, which is what the ROV team does. And you can then measure those abalone back in the lab if you missed them. And what has been generated over time is this length frequency distribution with total length here on the x-axis. Oh, sorry, this is density. Oh, sorry, it's length plotted by density, sorry. And what you can see here is that these lengths over time have been moving to the right. And if you take a look at the information from 2014, you can see that there are very few animals in the smallest size classes. And this really does concern us. And we now know that at this population, we've got a declining population, and it's declining by about 12% per year. Okay, now on to density and population size. So we do see differences across the different depth stratum based on the ROV surveys that have been conducted from 2002 to 2014, with this middle depth zone seeming to be the area where white abalone occur at highest densities. And here is the population estimates for these same data over time. And you can see here, if we just focus on the middle distance, that we have seen a decline in density and a decline in abundance over time. And here's a table that demonstrates the population estimates from 2002 to 2014, showing you that, yes, in fact, this population is declining at a rate of about 12% per year. So we don't have replacement of small animals in the smallest size classes, and we've got numbers and densities going down. We were also able to estimate nearest neighbor distance, which isn't really a true nearest neighbor distance when you're using the ROV, but it's the best we can do. And what you'll notice here is that many of the animals that we find with the ROV are quite far from one another. So this is distance here on the x-axis and the frequency here on the y-axis. So we've got quite a large number of animals that are very far apart from one another. And this is just another way of looking at the nearest neighbor data, where you can see here that the lighter colors are animals that are fairly close to one another, like the white dots. But as you get into the orange and yellow, those animals are more than 50 meters apart. Now, even though nearest neighbor distances are not where we think they need to be, we think abalone need to be very close to one another in order to spawn successfully. We have seen some animals in the wild that are in a fairly small size class, less than about 100 millimeters in size. And so if we use the only data that we have presented by Tutt-Schulte on age and length relationships, we do have an indication that some spawning may have actually occurred since the listing in 2001. But that spawning is very, very limited, and there are only a very small number of individuals in that size class, suggesting some of the animals may be as young as 4 years old, but we're really not sure. We have to take that bit of information with caution. Now, we've also used the ROV to explore other areas to see whether white abalone might exist in places along the mainland. And we have actually found some, which we're very excited about, but the numbers are very low. And again, we've done this with Pinto abalone as well. And again, we have very, very few animals that are relatively small. But using the ROV has allowed us to narrow our searches. So the ROV, as I said, you can think of it as a rover. It's doing a broad sweep of an area because it can cover a lot of ground. What we can then do is go to places where one white abalone is found and do a much better search using scuba. And so we started doing scuba surveys in about 2010, and over the last 5 years, we've actually found quite a few Pinto abalone and white abalone in the near coastal area. So we were very excited about this, and we wanted to learn more about these remnant populations for the reasons that I explained earlier. So here are some scuba results for white abalone. This shows you length of the white abalone that we found and the frequency of the animals. And again, most of the animals seem to be quite large, but there are a few animals in these smaller size classes. One of the most important things we learned was that we needed to visit sites repeatedly to actually find all of the white abalone that were in the area. And again, we're talking about a relatively small area. We're laying out transects that are about 50 meters long, and our divers are looking anywhere from 2 to 4 meters on either side of that transect. Well, covering that relatively small area, we had to do that 11 times in order to sort of level off with the number of white abalone we found in an area. And we dove that site enough that we became familiar with all of the animals. We, in fact, named them. So we're pretty sure we're not double counting here. So one of the lessons that we learned was, after you've done that broad sweep of a habitat, you really do need to send divers and spend quite a bit of effort and time searching an area thoroughly. White abalone are quite cryptic, even when they're large. Sandy can tell you that they're very easy to miss, even though they're out in the open. They're not a crevice-dwelling species, as far as we can tell, in Southern California. So they're out in the open, but they really look like rocks, and so much stuff is growing on their shell that they're incredibly cryptic. So you really do need to go to an area many times to be confident that you've got an accurate estimate of abundance. We did just that in one area where we happened to find about 9 white abalone. And just to demonstrate to you how we can then calculate nearest neighbor distances is you can lay a grid over this area you know you've surveyed, and you can come up with an area that you've surveyed and a density, and you can also calculate nearest neighbor distances because you've really searched that area. And what we found, at least with white abalone in this one small area, was that Some of the animals were within 5 to 6 meters of one another. Quite a few of the animals were within 10 meters of one another. And then what about this poor guy out here? He seemed to be about, whatever that is, 60 to 62 meters from his nearest neighbor. But again, nearest neighbor distance is really just an estimate of nearest neighbor distance, because his nearest neighbor might have been a few meters away in an area that was off of our grid. So you have to take these estimates as just that, estimates. We have a partner who we work with in San Diego. Her name's Amanda Bird, and she's a graduate student at Cal State Fullerton, and she's done her master's thesis on Pinto abalone, doing very much the same thing, where she does roving surveys, has found Pinto abalone, and then she does concentrated surveys in a relatively small area of the ocean floor. And she also divided her surveys up by depth stratum. And what she found out about Pinto abalone is that they like this mid depth stratum between 21 and 30 meters. She found a much broader distribution of sizes of Pinto abalone, including some Pinto abalone that were very small. So it really challenges our understanding of what that nearest neighbor distance needs to be for our abalone species, because it appears that we've got some recruitment going on with Pinto abalone, but we still have densities that are well below what we think, or well above what we think they need to be in order for reproduction to be successful. So Amanda believes that aggregation size might be a more appropriate metric to measure for abalone, because it may not matter how far an individual abalone is from the nearest neighbor you can find. What might be more important is what the aggregation size is and what the ratio of males to females in that aggregation size is. So she's playing around with collecting some different metrics in order to help us figure out what the most important metric is for gauging abalone status. Okay, so now on to some results from the in-situ time-lapse camera. This is going to be fun. This is time-lapse video of a pair of white and Pinto abalone on the same rock. And I'll let you just enjoy the moment of looking at these abalone cruising around. The field of view for the in-situ time-lapse camera is about 1 meter. So we are really at the finest scale you might imagine for observing an abalone here. And take note of the fact, this is roughly 2 weeks of video compressed down into this little movie that I'm showing you. And I want you to just notice that there's basically a large amount of food in every frame. And these abalone are eating quite a large amount of food. This was just before the temperature started warming up this summer. So there was still a lot of kelp around. And that little foray off the rock was the only foray out of the field of view that occurred over this 2-week period. So what are we learning from time-lapse camera deployments? Well, first of all, we have learned that you can deploy these things almost continuously for up to a year now. We've had these cameras on the same, we have 2 pairs of Pinto and white abalone that we've deployed these cameras on. And they spend very little time out of the frame. So we're thinking, in large part, these animals haven't moved for about a year, except for the movement that they make when they're eating. But it is pretty clear food was abundant. So the next step for this work is to really try to match up this visual data with some of our environmental data and try to figure out, when are they moving most? What really is in the video? What other visitors are moving in and out? You might have seen that chitin that kind of cruised across the frame. Who would have thought that a chitin would move faster than an abalone? But anyway, there are many different things that we can learn by looking at these videos more carefully. And so we've got a Coast intern who's going to be working with us this summer on analyzing these videos. Okay, and now I'm going to talk about the last tool that we've been using to learn more about abalone in the wild. And this is the movement study that we're doing with Pinto abalone using the acoustic tags and the acoustic receivers. So this is just a plot that shows you our area where we deployed our receivers. These little stars of David here are our receiver locations. So we have 7 receivers deployed within this 100 by 70 meter area. And the black squares are all of the Pinto abalone we tagged. We tagged 25 Pinto abalone. And the white diamonds are a few white abalone that we happened to find in the area. We did not tag them, of course, because we don't have a permit to do it. But we did note their presence, and we did measure nearest neighbor distances for them. And now I'm going to show you another movie, because movies are fun, of one of these Pinto abalone getting a mock tag. We were concerned about what tagging these animals would do to their behavior and whether or not it caused any mortality. So we mock tagged a couple of abalone. Sorry, hope nobody gets seasick out there. We mock tagged a couple of abalone, and then we put the in situ time-lapse cameras on 2 of the Pinto abalone that we mock tagged, and we tracked them for 2 weeks. And what I can tell you is that they too did not move out of the frame of the time-lapse camera. So, and it did not appear, at least on first review of the movies, that this tagging process did anything to alter their behavior dramatically. So we're pretty confident that this could become a very useful tool for not only figuring out what remnant populations are doing right now, but in the future, as we select sites for outplanting and move forward with our outplanting efforts, how are we going to track these animals over time as they grow? Well, this might be a very important tool for learning how to do that, or learning what kinds of densities we should be putting out in nature. So we can learn all kinds of things from this work. Here is some information on the Pinto abalone nearest neighbor distances we've been able to generate from this work. And you can see here that we really had what we were looking for in this population of 25 or so Pinto abalone that we tagged, in that we had quite a few abalone that were sort of off here in the distance, very far, over 12 meters from their nearest neighbor. But we did have some groups that were very close to one another, so we had a nice broad distribution. And I also included some of the nearest neighbor information for the white abalone in this area, mostly just to point out that the 4 white abalone in this area are all greater than 10 meters from one another. So we're not sure whether this is a viable population or not. They're not that far from one another, but they're farther than what the conventional wisdom would suggest to abalone need to be in order to spawn successfully. So, in summary, we are learning so much from all of the work we're doing in the wild. Our captive propagation program, of course, is hugely important, and the aquarium is a place where you can see white abalone. It's one of the only places you can see white abalone in Southern California. So I encourage you to do that. The work that we're doing in the field complements everything that's happening in the lab. It's preparing us for what happens next, and it's preparing us for how to proceed with outplanting. So between ROV, scuba, time-lapse cameras, and acoustic tags, they each can provide a unique bit of information that will guide our recovery efforts, because the type of monitoring that they allow us to do are all different, and they occur at different scales. We know that the monitoring we do should probably take 2 forms. We don't have a lot of time to search for what seems like a needle in a haystack, so we've got to do that broad sweep and cover a lot of ground. But then when we find something interesting, we need to hone in on it, because it could be that after much effort at that site, we'll find more cryptic abalone there. While what we know at this point about size-age distribution suggests that recruitment has been very limited, we still need to look at that more closely and probably generate better size-age relationship information. So we really can say something about, approximately what age is an animal that's 100 millimeters in size. We really can't say that with much certainty right now. Even if there is some natural recovery occurring, it is extremely limited, and it may only be occurring in a tiny number of places compared to the historic distribution of both of these species. So it's important that we continue to monitor, but we can't just do this. We have got to do something to enhance these populations, and we need to do it now before the PDO ends, if it hasn't already, because conditions have been very good and favorable for abalone over the last decade with relatively cool water. So we have to do something. They're not going to recover on their own because in many places they are locally extinct. There are so many areas where we need additional studies. We really need to better understand these early life stages of abalone. We can do that in the laboratory, and it's being done right now. But we really should be thinking creatively about how we can address this in the wild as well. And we do have investigators who are thinking about this very thing by collecting bits of coral and rock and bringing it back to the lab and looking for just settled abalone under the microscope. Cal Fish and Wildlife is doing that work. They're also trying to track recruitment using these modules that they have placed along the coast to try and find baby abalone. But we need to do even more because we really don't understand how these phases of life are working. And let's see, what else? Did I mention genetics? Genetics is also a really important area for future research. We need to understand what kind of diversity we have out in the wild so that we can try to replicate that in the lab with our broodstock and make sure that we're raising captive abalone that are as diverse as they can possibly be so that when we do outplant them, they've got a better chance of surviving through all of the challenging times ahead. So with that, I'd like to acknowledge some of the primary people that I worked with in putting this talk together. Kevin Stierhoff, Dave Whitting, Bill Hagee, John Williams, and Amanda Bird, and also all of our abalone partners in Southern California and Northern California have made this program really, really, really exciting. So thank you.